Hello friends, in this video we are going to understand about different stages of Euglena viridis. Locomotion, nutrition, respiration, excretion, osmoregulation, reproduction, encystment, sensitivity of Euglena viridis. Motion of Euglena viridis. In Euglena viridis, motion is affected in the following way. These are the types of locomotions. Euglena viridis, types of locomotion. Euglenoid movement after Butcher's bomb. Then B is rowing by flagellum. Flagellum assisted locomotion. The actual mechanism involved in flagella is not satisfactorily known and there are varieties of flagella movements. It has been advanced to explain the forward motion that the flagella makes a series of lateral movements and as a result exerts pressure on the water at right angles to its surface. This pressure is resolved into two forces, one acting parallel and the other at right angles to the axis of the body. The parallel force causes the body to rotate, while the force acting at right angles moves the animal forward. Another observation states that Euglena viridis moves by the wavy motion of the flagellum. A series of wavy waves pass along the flagellum from base to tip at a rate of 12 per second, which pushes the animal forward. Flagella action exerts a force on the surrounding medium which moves the water away from a stationary animal. The waves move in a spiral manner along the flagellum and cause Euglena's body to rotate once a second. Thus, in its motion, it traces a spiral path around a straight line and proceeds. The rate of motion is 0.5 mm per second. Rowing. In normal locomotion, Euglena viridis can also walk by rowing. When rowing, the beat of the flagellum has an effective stroke and a recovery stroke. During effective stroke, the flagellum is held rigid and slightly arched in the direction of the stroke. Effective stroke helps to push water backwards and body pulls forward. During recovery, the stroke flagellum is strongly curved and the flagellum is brought to its normal position and minimal resistance is encountered during recovery stroke. Euglenoid movement. Euglena sometimes shows a very peculiar motion in which waves of contraction move along the body from anterior to posterior end and the animal crawls forward. The contraction is brought about by the stretching of the protoplasm on the pellicle or by filaments localized in the ectoplasm called myonemes. Nutrition in Euglena viridis. The modes of nutrition in Euglena viridis are holophytic and saproroic. Like a true plant, it assimilates carbon and forms carbohydrates from carbon dioxide and water. The holophytic type of nutrition occurs in the presence of sunlight, and the green pigment chlorophyll plays an important role in the process. Nitrogen and other minerals that remain dissolved in pond water are absorbed by the cell surface. The excess manufactured carbohydrates is stored in the form of paramylum. Euglena remains an autotroph as long as it is exposed to light and is provided with essential inorganic compounds. The entire autotrophic process in Euglena depends on external sources of vitamin B12 synthesized by bacteria and some microorganisms. Sometimes when pond water becomes polluted with dead and decaying organic matter, Euglena leaves the holophytic mode of nutrition and switches to a saproroic mode. The dead and decaying substances dissolved in the pond water and are digested externally, and then they are absorbed through a normal surface of the body. Some workers have reported that movement of the flagellum forces small organisms to enter the reservoir and are swallowed. Such occurrence of the holozoic mode of the nutrition in Euglena is open to doubt. Respiration in Euglena viridis. Respiration in Euglena viridis is aerobic. It absorbs the dissolved oxygen from the surrounding medium by diffusion. The process of photosynthesis releases a good amount of oxygen during the day. There is very reason to believe that this oxygen is used in metabolic activities. Excretion in Euglena viridis. The carbon dioxide accumulated in the process of daytime respiration is used. In photosynthesis, unused CO2 carbon dioxide is removed by diffusion through the surface of the body. Nitrogenous waste products are also removed in the same way. Osmoregulation in Euglena viridis. Excess water is removed by contractile vacuoles and its tributaries. The radiated or associated small vacuoles collect surplus water from the endoplasm and release their contents into the main vacuole, which gradually increases in size and eventually contracts to force fluid into the reservoir. The fluid from the reservoir drains out through the gullet. Along with this, water-soluble wastes get out of the body. Reproduction in Euglena viridis. The common mode of reproduction in Euglena viridis is longitudinal binary fission. The producer daughter cells are mirror images because the division is symmetric. This diagram shows events during longitudinal fission in Euglena. During fragmentation, the locomotive activities are suspended, and in some cases, the flagellum is retracted. The blepharoplast first divides, and the two halves are connected by a spindle-like structure or a strand. This is followed by the eumeiotic type of division of the nucleus. From the reservoir, the cleavage group becomes visible and proceeds longitudinally to divide the animal into two halves. The lost parts are regenerated soon after the Euglena splits into two daughters. Each one develops a new flagellum. In some cases, one of the daughters retains the flagellum of the mother, and in other, a new one develops. Sometimes several Euglena clumps together lose their flagella and become rounded. They secrete sticky substances in which they are embedded. This stage, called the Pamela stage, is often seen as green scum on ponds. Individual members of Pamela carry out metabolic activities and reproduce by fission. When favorable conditions return, the Euglena detaches, regenerates the flagellum and begins to lead a normal and active life. Encystment in Euglena viridis. Euglena viridis encyst during periods of drought and extreme cold. The animal becomes inactive, withdraws the flagellum and assumes a rounded shape. Gradually, the protective walls are secreted. The cysts are red due to the presence of a pigment called hematochrome. These two diagrams shows Pamela stage of Euglena, encysted Euglena undergoing division. On return of the favorable position, the cyst wall breaks and the euglena comes out. Nuclear division can occur in an encased euglena. Sensitivity of Euglena viridis. Euglena viridis shows photosensitivity and their responses vary according to the intensity of the light source. Normally it floats parallel to the light rays and towards the source of the light. The stigma with thickenings on the flagellum forms a kind of optic organelle for the animal. The animal can also respond to different concentrations of chemicals, oxygen and carbon dioxide.